is Tyler Mominey. I am the Associate Coordinator here at DEP's Coral Reef Conservation Program. I'm based in Miami, Florida, and one of the numerous responsibilities I have is overseeing our 2 citizen science programs, including CFAN and BleachWatch, which is what you're here for today. Just kind of before we get started, and as people are still joining, my colleague Rachel Scooble is going to be joining me in helping to moderate and keep track of all questions. I'm actually going to pause at some sections for questions. So keep track of your questions. We'll try to get them all answered. Worst case, if we can't answer them, we'll go ahead and just email you a definite answer. Also want to point out that this webinar does include some PDF attachments. Normally, when we do these in person, you would get a folder or a USB drive with all of them. I do want to point out that they are on our website. We're also going to send out an email post-webinar that'll include all of those, and just to make sure that you have all the documents that you need to be a BleachWatch observer. So first of all, just thank you for your time. And you know, skip slides here. Florida's Coral Reef. So Florida's Coral Reef is obviously what you guys are here to help protect, and appreciate that. And while starting off, I just want to play this introductory video to kind of get a little bit more information on Florida's Coral Reef.
A living kingdom, a refuge, and all of it a gift. We will protect its splendor and mystery. We will preserve the gift we have received, our inheritance, our legacy.
Awesome. So that's a little bit of background, kind of inspirational about Florida's Coral Reef. And now that we know a little bit more, we're going to start off with what is coral bleaching? Now that we have some context, and this is kind of our agenda for today. So we have what is coral bleaching, then we're going to go into coral disease in Florida, CFAN, BleachWatch, which is going to be your training, and then furthermore, your contribution. And again, we're going to stop for questions, and you're also welcome to email me, and I'll leave up my email at the end of this to any questions that you may have, and even throughout the process of putting in BleachWatch observations, anytime you're able to reach out to me and our team and just get some more answers. We're going to move forward with what is coral bleaching. Apologies for the delay. Computer's running a little extra slow today, but that's all right. First off, we're going to go over some coral anatomy 101. So there are 2 categories of corals that we're going over today: stony corals and soft corals. For this class specifically, we're going to be focusing on stony corals. So corals are the animals. They are animals. Not everyone knows that. Some of you may know that. And they're actually in the same family as jellyfish and anemones. But they actually have a plant and mineral component, so it gets a little confusing. And we'll tell you more about that in a little bit. These animals secrete a hard limestone exoskeleton, which is why they're called reef building or stony corals. What we're used to seeing, kind of like in this photo, is a coral colony, which is made up of numerous individuals called coral polyps, which is that nice little picture in your top right. Get my pointer working right here. So that's what the coral polyp looks like, and we're going to get into a little bit more of what the polyps actually do. So coral anatomy, continuing on. And if you look at this cross section of the coral polyp, we can see there's this hard limestone skeleton, which you can see in kind of that little breakout right there. And that provides the coral structure. This skeleton is actually made out of calcium carbonate, which is the mineral part of the corals. And you can see that better in this picture right here. So you have that calcium carbonate, which you may or may not have seen, but that's what some people think of as coral when they think of it. Now, covering this actual skeleton is a soft tissue, and that soft tissue forms a polyp with tentacles, as you can see right here, and that's actually surrounding the central mouth. Each of these polyps are secreting that white limestone skeleton that we saw first, and there's actually a variation of coral that you may see out there on Florida's coral reef. And they don't come just in one shape and size. As you can see, you kind of have these long branching fingering ones, and also maybe you're familiar with these brain coral here. And we're going to go into that a little bit later and how to differentiate throughout the corals. Coral feeding. If you look closer at the tentacles, this is where the corals actually feed in two different ways. The first way is that they're filter feeders. So on the outside of their tentacles, they have stinging cells, which they use to capture plankton from the water column at night. And this only provides about 5 to 10% of food and their nutrients. So you actually might be asking yourself, how else do they feed? Because if I only ate 5 to 10% of the food I needed, I'd still be hungry. Now that second way is corals actually have a mutually beneficial relationship with a microscopic algae called zooxanthellae located deep inside their tissue. This mutually beneficial relationship is called a symbiotic relationship, since both of the coral polyp and the zooxanthellae benefit from this relationship. This symbiosis is where the coral contains a plant-like component. And that zooxanthellae is kind of a hard word, might be a hard word to remember. So a good way to remember it is zooxanthellae lives in the belly. And that's how you can remember that. These are where they eat. Now, the zooxanthellae are important for two reasons. The first reason, that the zooxanthellae actually provide 90 to 95% of the food and nutrients that coral needs to survive. And the zooxanthellae photosynthesize, meaning they convert carbon dioxide and energy from the sun into carbohydrates and oxygen that the coral needs to survive. That second reason is that zooxanthellae are also responsible for the normal healthy color of coral. Living coral tissue is actually clear. If you can see the picture of the coral polyp, all these little tiny golden, almost looks like pollen, the specks are zooxanthellae, which give the coral polyp the overall kind of golden color, and there's some variations on that you'll get into. So like I said, corals can come in a range of colors in Southeast Florida, and they're usually earth-toned, not a definite, but either way, you can't see that white skeleton underneath it because of the algae in the tissues, which is giving it that color. This is actually an even better close-up. You can see that translucent and then that zooxanthellae right there. And in places where the zooxanthellae are present, the tissues appear colored, kind of like I was saying here with those brown little specks. In places where there are no zooxanthellae, the tissue appear clear and translucent. Towards the bottom of the photo, right here, you can kind of see that white limestone skeleton of the polyp underneath the translucent tissue, where there's actually no zooxanthellae present. Well, healthy corals are composed of numerous individual polyps, like we've been talking about, each of which contains zooxanthellae within their tissue. When the coral polyps become stressed, they will expel the zooxanthellae. And without the zooxanthellae, the coral polyps lose their coloration. What's left is the clear living coral tissue covering their white limestone structure. And because the corals turn from being colored to appearing white, as you can see here, that's where we got the term coral bleaching. Now, coral bleaching is a natural response, and it can occur from a number of environmental disturbances. And maybe you've heard of some of these stressors before. You can think of some off the top of your head. Some of the few that you might think of are pollution, oil spills, low oxygen in the water, extremes in solar radiation or sea temperature, changes in salinity, sedimentation, or disease. And coral bleaching is actually a gradual process. Here you can actually see the progression from a healthy to a paling, which is kind of that intermediate phase. So there's some color and some white patches, to a completely bleached coral. That's not what we want to see, but we may see out there. Now, I have an important question. Is a bleached coral a dead coral? A bleached coral is actually not a dead coral. And some of you got that right, and it is a little confusing. So bleached corals are still living, and that's because they still have live tissue. However, the zooxanthellae have been expelled, and therefore that's why the coral has lost their color. Another way to kind of think about it is being in an ICU in a hospital. The coral is under a lot of stress, but it can be brought back to full health. So if the stress is removed and then the environmental conditions return to normal, soon enough we can actually get a healthy coral, and that zooxanthellae can come back and they can actually survive and look nice and colorful just like that. This typically takes about a couple weeks to months for the corals to regain their zooxanthellae, and it just varies based on the species of the coral. Unfortunately, if water temperature increases, it only takes a matter of days to weeks for a coral to bleach. And furthermore, if those stressors continue and are severe enough for an extended period of time, it can lead to total mortality, which is not what we want. Now, the mysterious question we have with the bleached coral is, will they regain their zooxanthellae and return to the healthy condition and survive, or will the coral unfortunately pass if those stressors persist? In other words, is the coral resilient? Now, just a quick definition: resilience is the ability to either withstand an impact and not bleach at all, or recover when it does happen. All right, apologies everyone again. So again, we are questioning in the BleachWatch Observer Network overall, is questioning really, is the coral resilient? And that's what you all are here to help us figure out. Whether or not the coral recovers from bleaching, there are some noticeable long-term effects. Since the coral have lost their primary food source, they have less energy available for skeletal growth and reproduction, but their overall health has been compromised, leaving them more susceptible to things like disease, predation, and death. Ultimately, dead corals results in the loss of habitat for other creatures for the reef. Here's a nice infographic from National Geographic here on your left, showing the history of mass coral bleaching events. Now, the first widespread bleaching we ever saw was from 1982 to 1983, right here. And it was associated with an El Nino event. And then we have global bleaching events in 1998 and again in 2010, which was actually a cold water event. The longest event ever observed was not too long ago, from 2014 to 2016. With back-to-back years, it means corals didn't have enough time to recover and before they were hit again with more intense stress. And we have some photos of bleaching, so you can see it's across different scales, and it can be broad sections of the reef like the top two pictures, or it can affect individual coral colonies, as you can see in the bottom two pictures. Now, the most important thing to remember is that the tissue's still there. Coral polyps are still alive. They just have no zooxanthellae, in the sense that they are starved. We have some more bleaching examples. I also want to point out that it can vary at all scales by region, reef, site, species, and colony. It may be due to a variety of reasons that cause one coral to bleach over another, as you can kind of see in this picture. Same water column right next to each other, and one bleaches and one doesn't. And this can be due to a variety of reasons, from exposure, depth, location, currents, that might be cooling and not one on the other. So that's why the corals in this photo, which are adjacent colonies of brain coral, one, as you can see, is bleached and one is healthy. And here, to make things even more complex, it's not as simple as white is bleaching. There's one species that we see here in Florida, the massive Starlight Coral, which is a pure fluorescent blue, purple, or pink when bleached. So it's not just the white. And that's because this species in particular has pigments in their tissue. It happens to look like an encrusting sponge. So just something to keep in mind. So I'm gonna stop here for questions. How do you guys feel about bleaching? Is there any questions thus far? All right, so we're gonna keep moving forward, trudging along. And with that, we have another poll, if it will let me relaunch it. Maybe not. So the question is this bleaching or not? I'm not able to reshare the poll, so that's okay. So just note it in your head, if this is bleaching or not. We're looking right here. So to answer that, no, this is actually not bleaching. Branching coral actually have fast growing tips where the zooxanthellae have not caught up yet. So this is not bleaching. To confuse you a little bit more, we also have predation. This might look a little bit like coral bleaching, but if you look closely, you can actually see part of the coral base has been eaten away, and the tissue is gone. Maybe this one here is kind of a good example, and you can see everything is completely gone. So parrotfish and damselfish, they'll leave those small circular lesions, in the coral.
Sorry, Tyler, again, I think that the slide has not advanced. So we're still staghorn. We might have to,
yeah. I'm gonna do the sharing again.
Okay.
How we look?
Looks great, yeah.
Okay, perfect. So we're on fish bites predation, correct?
Correct.
All right, perfect. Thanks, Rachel. So parrotfish and damselfish, they will leave those small circular lesions, which you can now see, those top pictures, which could be mistaken for bleaching. And fireworms, the bottom two pictures, leave trails of missing tissues when they feed. Moving forward here. So now that we've done a little introduction to coral anatomy and bleaching, I just wanted to ask some quick questions to review the content that we learned. So we're going to launch a couple polls here, gauge your knowledge thus far. So first and foremost is, how do corals feed? And for this one, there can be more than one right answer. So corals can actually feed through a symbiotic relationship with their tentacles. So the correct answer is A and C. Got another one for you. So true or false, a bleached coral is a dead coral? All right, perfect. So all of you answered correctly. False. A bleached coral is not a dead coral. So I'm glad that you're all paying attention. One more question for you. Where do reef building corals get their color? All right. So it's the zooxanthellae, answer B. Remember, zooxanthellae lives in the belly, and that is where the reef building or stony coral get their color. So moving forward, I'll take a couple more if there's any questions and just stop here, give you all a breath. I have one question. Are parrotfish and damselfish native? So yes, there are some species of parrotfish and damselfish that are native, and they just happen to feed on coral. All right, there's no other questions. I'm gonna go ahead and move forward with coral disease. Now, there has been a recent, semi-recent outbreak in Florida, which has actually sparked the need for us to expand our response in our BleachWatch program. First off, what causes coral disease? Coral diseases generally occur in response to a microscopic organism such as a bacteria, virus, or a fungus. And the origins and mechanisms of diseases on corals are still not very well known, and it's difficult to determine how these biological responses occur. Things such as sewage input, pollution, or other environmental stressors are often the root cause, but again, it's kind of difficult to determine. Coral bleaching can actually also leave corals in a weakened state, as we kind of just brushed over, which will actually make them more susceptible to infections from coral diseases. So black band disease, yellow band, and discolored spots appear as spots or lesions on the surface of coral. Over time, these lesions progress across the coral surface, consuming the living tissue and leaving the stark white skeleton behind, which you can kind of see in some of these pictures. Other diseases like white band, white plague, white pox, and the newer one, stony coral tissue loss disease, which you may or may not have heard of, cause large patches of living coral tissue to be sloughed off, and they expose the skeleton beneath. In both cases, diseases are usually characterized by a distinct line between live and dead tissue, as you can see in these photos as well. And that's an important feature and distinguish between coral bleaching and coral disease. And there'll be some more pictures up ahead. Just want to make that note. So once the bare skeleton is exposed, it becomes the breeding ground for algae and other encrusting invertebrates. So as you can see, kind of that green and kind of fleshy color at that top and before the lesion. Now, to help you recognize coral disease and think about it this way, many of you, if you're divers or snorkelers, even just free divers, maybe you've seen a can or other marine debris on the bottom of the ocean floor. And if you think about it this way, what's always on the outer surface? Usually it's algae. So if you think about that, you think about the cans that you might see at the bottom of the ocean. The first thing that's kind of going to catch on is that algae. Well, the recently exposed skeleton, it will fairly quickly be covered with a thin layer of filamentous algae and will stop appearing that bleach white. Once that happens, it really compromises the overall health of the colony. So here's actually a really great graphic of the polyps to indicate the difference. The yellow polyp here, it's full color, which means it's healthy. And as we move in the polyp in the middle, you can see it's gradually losing its color. But the polyp is in fact still intact, so you can see the polyp there. And the polyp on the right also loses its color, but eventually you can see that structure disappears because the tissue is completely gone. So again, the difference is bleaching, tissue still there, zooxanthellae are gone. With diseases, and especially tissue loss diseases, everything will be completely gone, leaving only the skeleton. So although coral bleaching and coral disease like I said, can cause a white appearance on the coral. There's a key difference to readily distinguish between the two conditions. The main difference in identifying coral bleaching from coral disease is that when a coral bleaches, it's still alive, so the clear tissue is still present, overlaying the coral skeleton. When a coral has been infected by a disease, the tissue itself will start to die, leaving the bare skeleton exposed. Eventually, this newly exposed skeleton becomes colonized by algae. So here, this one is actually not disease. I want to point that out, but it gives you an idea of what the bare skeleton looks like compared to the bleach polyps. So in this pink circle, it's that bare skeleton, and you can see the bleach coral up here in that yellow orangish circle. Now, stony coral tissue loss disease outbreak, which again you may have heard of, is somewhat of the most recent outbreak, and it is actually what caused us to expand BleachWatch to include disease starting in 2014, and it's still ongoing today. So talking about the disease progression, so it was first observed in the summer of 2014 in Miami-Dade County, just north of Biscayne National Park. In 2015, it started rapidly spreading north, first through Broward County and slowly south into Biscayne National Park. And if you recall, 2014 and 2015 were extremely bad bleaching years, so the corals were already in a weakened state. In 2016, it spread through Palm Beach County and into the Upper Keys. 2017, it spread across the entire northern portion of the Reef tract through Martin County and then into the Middle Keys going south. 2018, the disease continued to spread, making its way now to the Middle Keys. 2019, the disease spread mostly through the Lower Keys, leaving only really that Dry Tortugas National Park. 2020, stony coral tissue loss disease progressed through the Marquesas. And unfortunately, 2021, the disease has now spread to the Dry Tortugas, which means it officially is throughout the entire Florida Reef tract from Martin County all the way down to the Dry Tortugas National Park. Now, to date, we know that disease has progressed to various countries, island nations, and territories throughout the Caribbean. This includes, but not limited to, Jamaica, Mexico, St. Martin, U.S. Virgin Islands, Dominican Republic, Turks and Caicos, Belize, St. Eustatius, the Bahamas, Puerto Rico, British Virgin Islands, Cayman Islands, Guadalupe, St. Lucia, Honduras, and Martinique. We don't exactly know how it's being spread, but we do know that the disease is waterborne. We have some research and some people looking into ballast water as a potential vector for transmission, but that's just one of the many other methods that are being thoroughly researched. And if you look at this map, just our map from AGRA, the red points, stony coral tissue loss disease is present. Green points, stony coral tissue loss disease is not present. Purple is just a new submission that's being reviewed to see if it is present. In yellow, it may be present. So you can kind of see that widespread. Now, the types of corals being affected are our primary reef building corals that we kind of started to talk about. And some of these are now largely absent from sites, especially in Broward and Miami-Dade County, where the outbreak actually started. Florida's coral reef is an economically and ecologically important ecosystem, as you may know, and it drives South Florida's economy. Reef-related activities in Florida actually generate $6.3 billion annually and support roughly 71,000 jobs just in Martin, Palm Beach, Broward, and Dade and Monroe counties. The reef also provides some natural infrastructure which protects coasts from storms, provides habitat for fish, and many other important benefits. Because of the severity and unknown characteristics, and because we haven't been able to identify the disease agent, we had to give it a name, and it's different than any other disease because of the high prevalence, the high number of corals and species affected, rapid mortality, high rates of disease transmission, and the large geographic area. Now, due to the loss of tissue, it was deemed stony coral tissue loss disease, or SCTLD, which you will often see it named for short. Now, for context, background levels of disease are common for Florida. It's about the 2 to 3% range. But this current disease outbreak, as you can see, is about the 66 to 100% outbreak in some sites for some species. This particular disease is also extremely infectious. More than half, as you can see here, of approximately 45 species of Florida's reef-building corals have been affected. To give you an idea of the progression that I've been talking about, once a coral is infected, observations show that the colony will actually die within weeks to months. From these time series photos from the left to the right, you can see how quickly disease spreads across the colony in just 1 month. So we have January 5th on that left, January 19th in the middle, a couple weeks later, February 1st, we have almost the entire coral colony has been infected. Now, these 2 Endangered Species Act listed species are among most impacted by this disease outbreak, SCTLD, pillar coral and mountainous star coral. For pillar coral, which was already semi-absent along the reef, we've lost over 95% of our known colonies across Florida's coral reef. Fortunately, there were efforts to collect fragments of these known colonies and store them in a gene bank to later outplant back on the reef once this disease, and we kind of got it figured out and it settles down. Now, these landscape photos show numerous corals being hit by the disease outbreak, not just one here or there. Each infected colony is indicated by the yellow arrows. And not only are there stony corals being affected, but we're also seeing diseases in sponges and Gargonians too. So you can see here and here as well. Now, the question I'm sure all of you are thinking about right now is, well, what are we doing about it? What is everyone out there doing about it if this disease is really taking a toll on Florida's coral reef? So DEP is actually co-leading the response effort with FWC, NOAA, and the National Park Service, working with numerous partners from federal, state, local, universities, non-governmental, and most importantly, the South Florida community on a multifaceted response effort. We've established 10 teams that are focused on different aspects of research, like research, intervention, coral rescue, communications, data management, and restoration, just to name a few. We've also started working with some international partners. As we said, this has gone international throughout Mexico and the Caribbean, just because of the wide outbreak, and we want to coordinate a response. Now, CFAN and BleachWatch reports through citizen engagement have been paramount in understanding the spatial extent of the outbreak, as well as the different types of corals being affected. And if you have questions about disease response specifically, please send me a message at the end, and I can put you in touch with some of the disease response coordinators to answer your specific questions. All right, we're gonna run another quick poll just to make sure we're all on the same page. All right, now the average response is in. The answer is both A and C, the presence of live tissue and the sharp defined line. Now the color white can actually be associated with bleaching and disease, just something to keep in mind. All right, now before I move forward, any questions on coral disease or anything else? All right, seems like there's no questions thus far. We're gonna move forward with CFAN and BleachWatch. For some background, DEP CRCP is responsible for managing over 100 miles of coral reef, and we only have a staff of about 15 people. Although we'd love to all be out on the reef every day, it's physically impossible for us to know what's happening across the region every single day, which is why we rely on members of the community such as you to let us know if you guys are observing anything unusual, that is out there on the reef. So that's why we started this program called the Southeast Florida Action Network, or CFAN for short, to allow people to report those incidents so we can coordinate a response. I'm going to play another quick video just to introduce the CFAN program. Maybe.
Florida's coral reef is home to over 40 species of living coral and plants and animals. Healthy reefs shelter our shorelines from erosion and coastal flooding. The Florida Department of Environmental Protection is asking for your help to protect this vulnerable treasure. Report reef damage, marine debris, invasive species, coral disease, and bleaching to the Southeast Florida Action Network at cfan.net. That's SEAFAN.net.
Awesome. So I saw that there was a question of where to report it to. So you can report it to www.cfan.net/report, or you can call our hotline at 1-866-770-SEFL. That's 7335. So anytime you're out on the water, you see something. Got ahead of myself. If you're out on the water, you see something, anything occurring in the ocean, anything such as vessel grounding, anchor damage, fish kill, disease, marine debris, biological incidents, invasive species, even algal blooms, you can report it to CFAN. Now, CFAN is a little bit different than the BleachWatch program, as it doesn't require any special training. Think about it as the umbrella for BleachWatch. But today we're going to focus on one incident in particular, which is coral bleaching and disease. This program is a little bit more specialized, and you're all here to be trained in this specialty today. BleachWatch is a special branch of CFAN, which, like I was saying, it's under that umbrella. While anyone can report events to CFAN, including coral bleaching and disease. To become a BleachWatch observer, you must preferably be a certified diver and undergo the training, which you're all currently getting. At the very least, you should be able to be trained on this. Now, the reason we have this program is due to the fact that since the 1980s, coral bleaching has increased in frequency, both along the Florida's coral reef as well as worldwide, which has led to significant deterioration to our reef. So BleachWatch was created to help monitor the reef for coral bleaching both before it happens, during, and after a bleaching incident. This program helps us understand essentially where bleaching is taking place, what species are more vulnerable, how long and severe the event is, and ultimately what is the recovery and resilience potential of those reefs. This program was actually originally developed for the Great Barrier Reef in Australia. And it's since been adopted in the Florida Keys and used about 17 years. And then in 2013, CFAN BleachWatch was implemented on the northern portion of the reef. Remember, that's the area that we currently manage. And that includes Miami-Dade, Broward, Palm Beach, and the Martin counties. This area is managed by us, which is that red portion. And although these programs are separated by political boundaries, the two BleachWatch programs are essentially the same, and we work very closely with each other. Now, the goal of BleachWatch is to provide an early warning network to coral bleaching by monitoring climate and sea temperatures, involving the community, which is you all, in assessing the condition of the coral reefs, and combining all that information to produce summaries of the current conditions in the region, which are sent to anyone who's interested, but also to resource managers who might be able to use that information for decisions and other purposes, as well as you all, so you understand what you may be seeing out there. I'm going to briefly discuss the environmental monitoring side of the program, which is something that I do as the program coordinator. Then we'll get into your specific involvement in the program. Environmental monitoring. So environmental monitoring involves sea surface temperature or SST data from a program called NOAA Coral Reef Watch. Basically, we know corals are more likely to bleach if the waters are warmer than usual. And this map shows areas where the SST is higher than usual, which we call hotspots. The take home here is that anywhere that you see yellow are here on this map are areas where the water is at least 1 degree Celsius warmer than usual. Science has shown that the corals start to become stressed in these conditions above that 1 degree Celsius threshold. The coral bleaching risk increases even more if the temperature stays warm for a long period of time. Using this very interesting unit called the degree heating weeks. Take home here is if the temperature is elevated slightly for 4 weeks, significant coral bleaching is likely. If temperature is elevated slightly for 8 weeks or more, you can expect widespread bleaching and mortality. So based on how warm the water is and the length of time the water stays warmer than usual, NOAA issues alert areas, which include 5 categories based on how likely bleaching is to occur. In this map specifically, white is no stress, no bleaching is likely. The bleaching watch, which is that greenish area on your screen, kind of depending on your screen, the water temperature is only elevated a small amount, so kind of keeping an eye on it because corals might be stressed. Still below temperature when they usually get stressed, but important to keep in mind. Orange in this map is a bleaching warning. Waters have passed that 1 degree Celsius threshold, so we can expect that corals are stressed and bleaching is possible. The lighter red in this map is a bleaching alert level 1, which means coral bleaching is likely, and then dark red is bleaching alert level 2, which is widespread bleaching and mortality is likely. So this is really just a nice way to summarize the information. But remember that in Florida, most corals are really only located right along our coast. So that was enough on environmental monitoring. Let's get into how you can participate yourselves, which is why you're all here today. All of that environmental monitoring we just talked about, that's handled by the BleachWatch Program Coordinator, and we're just going to move into what you all are going to be doing. So in this photo are all the training materials that normally I'd be handing out to you in person in a folder or copied to a hard drive. Now these materials are either going to be emailed to you or we're going to find another way, and they actually are all available on our cfan.net/bleachwatch, which you can see at the bottom. You want to go ahead and access them beforehand before we get those over to you. And these include program overview, bleaching and disease fact sheets, the data sheet, which you can use to report your observations, data sheet instructions for how to fill out your data sheet, the Coral Condition ID guidebook, which can be printed as a booklet, so you can take it with you on the boat. But again, it's always readily available if we take some time to get to all your emails at cfan.net/bleachwatch. And we also now have dive slates with data sheet on it. And this can be hooked to your BCD if you're diving, or hooked to yourself if you're snorkeling. After you submit your first report, we can send you a dive slate if that is advantageous to you. All you have to do is just note on the BleachWatch report that it's your first report, and then we'll get in touch to you about how to get you the dive slate. So moving forward, the most important thing here, which is the data sheets. This is what you all will be using when you're out in the ocean and observing. We're going to go through this section by section. So section A, your observer details, which is the date and time of the visit, which is actually the most important thing because we use that to look up environmental data and track it over time. Contact information, name and email, always required. Phone number is optional, just in case we have to get in contact with you afterwards. The organization or vessel information. So did you go out with dive charter, company, or as a organization, the type of observer, whether you're a resident, visitor, you're in the commercial sphere, anything like that. It just helps us categorize where most of the information is coming from and where to focus for future years. But this, again, just helps us keep track of the types of people filling out reports. Section B, obviously we would love to get GPS coordinates just like that. I know it's not always the easiest, but if not, you can always put the site name or description. For example, Lauderdale by the Sea, we all kind of know where that is, or a street name, which we can look up in a map. Next, we have the depth range of the site. After that, the county, which just helps us categorize the observations regionally. And the environmental conditions, it's optional, but it is honestly very helpful if you can, or if you have the equipment that can include wind speed, air, sea surface, and bottom temperature, underwater visibility, cloud cover, all those things, they can actually influence the likelihood that bleaching will occur. So this, again, just helps us track when bleaching occurred, and we can reference this going back. Just remember when we're talking about different types of stressors, the corals can be stressed by bright sunny skies and clear calm waters. All right, moving forward to the next section. So did you observe signs of bleaching or disease? If you don't see bleaching or disease in their swim, that's awesome. We hope to see a lot of that. You're finished. So it's even easier for you. It's good for us. But we still do want you to report no disease because that's actually just as important to reports of disease or of bleaching because that helps us identify areas that might be more resilient to others and just helps us observe where bleaching is and isn't occurring. But if you do see bleaching or disease, then you're going to continue on to the next section, which is going to look like this, section C, the bleaching and disease observations. It's where you're going to indicate your observations for coral bleaching and disease for each type of coral. This section becomes extremely useful if you have the underwater slate that we were talking about earlier, where you'd actually be able to tally within each box during your dive. Before we get into the bulk of the section, you should first review the 6 different types of corals. Now these, just for your reference, come from Paul Heuman's book, Reef Coral Identification, which I highly recommend if you want to learn more. It's helped me learn a lot. Anyways, so if you're a more experienced observer or if you just want to kind of test your coral knowledge, we encourage you highly to identify it by family or genus and or species if you want, where possible in the notes section, but it's not necessary. This just helps us again to track it over time, which corals are bleaching and or diseased. Now, some species are more resistant to bleaching than others. So again, this just helps us to monitor. Now, the following 6 slides are going to show examples in each group from least common in Southeast Florida to most common. So right here we have the flowering and cup. So you can see the coralites appear independent from the rest of the colony, almost like a bouquet of flowers. This specific one that you're seeing is the smooth flower coral. You may see this in a gray, a yellow, to even a blue or purple. Next up, we have fleshy coral. Now these have a fleshy or fuzzy appearance. These include, as you can see, cactus or mushroom coral. Now some people do get confused with brain coral and fleshy coral, but these specifically look soft and fuzzy. That's how I like to think about it, but please be sure not to touch it. Now, there's 4 species total. They are fairly uncommon in Southeast Florida, but they are more common in the Keys. And they come in, as you can see, a wide variety of colors. Next up, we have plate, leaf, and sheet coral. These are flattened or layered. They grow in thin sheets or flattened plates. There's 3 species really, Only 2 of them you're probably going to see here in Florida. So we have lettuce coral and the white star sheet coral that you will most likely see. That white star sheet, those noticeable white dots, that's actually part of the coral, not bleaching or disease. Next up, we have branching and pillar coral. Like the name suggests, these are the ones with noticeable branching or pillar shape. We have elkhorn, staghorn, finger coral, and pillar coral. 5 species total in Southeast Florida. Now, elkhorn, staghorn, and pillar are all on the Endangered Species Act threatened list. Next up, brain coral, which you all may be familiar with or have seen already. These are corals with brain-like grooves and are usually round or massive. There's a total of 5, but 4 the most common that you'll see in Florida are symmetrical brain coral, the groove brain coral, the maze coral, or the Boulder brain coral, or the knobby brain coral, which is the least common of the 5. Next up, we have the encrusting mound and boulder. Now these take on the shape of what they grow, and they're usually pretty massive or round, excluding the brain coral. And these are by far the most common you're going to see in Southeast Florida. Maybe you've seen them already. We have 10 species. Almost all of them have star in their names because of the star shape of their polyps. So we have lobed, boulder, and mountainous star coral. We have great star coral. We have smooth star coral, the mustard hill coral, and the massive starlight coral, which you can see all on this slide. And just to keep in mind, I know this is probably overwhelming, but we do have kind of a coral cheat sheet that would be in the materials that we will send all of you. So don't feel like you have to remember all of them right here, right now. We're going to go back to section C on that data observation sheet. Now that we know the types of coral, we're going to start on the bleaching side. Here you're going to mark the severity of bleaching for each type of coral. This is where it becomes extremely helpful to have the slate because you can mark them underwater. Right here we have 4 categories: no stress, paling, partial bleaching, and bleached. Paling is the initial stage of coral bleaching, so they're starting to lose their color. This is actually the first picture I kind of showed you, which I'll show you again on the next slide. So they're starting to lose color, or very light or yellowish brown color. I'm sorry, yellowish color. Then the partially bleached is only really on the upper surface. There'll be patches of fully bleached or white tissue. And of course, the totally bleached is no color left. And again, corals can be tricky because they come in lots of different colors. But a good tip is to look at the colors of other corals of the same species around them at the dive site just for comparison. So we're going to look at some pictures of paling here. As you can see, there's a noticeably lighter pigment. The polyps are just starting to bleach or they're recovering from bleaching. Close-up picture of paling really emphasizes the point. You can see that the tissue is still there in some of the polyps, but the color is gone. In others, it's starting to fade. So that's paling. Moving forward, partial bleaching, we have patches of fully bleached or white tissue. On the left is that great star coral. We have lettuce on that top and groove brain coral, so all look a little different, but that's partial bleaching. Some more pictures where some areas are completely bleached, but the others remain a normal coloration. And that bottom picture, if you look at it, now that I'm looking at it, there actually is partial bleaching and some paling as well. Now, the top one, as you can see, is a little weird looking. That's because the massive starlight coral actually looks fluorescent pink when bleached. And moving on to fully bleached. So the tissue, again, is totally white There's no zooxanthellae present. And remember, just you're looking for that clear tissue. You can kind of see it there. It can be more difficult to see on certain species, but do just the best that you can, obviously without touching them. It can vary. Just look at some other corals around you to compare the colors, just as a gut check to yourself. Now we have in the top right, the bleach next to a perfectly healthy groove brain coral. In this picture, you can see that stark bleached white staghorn coral. And here we can see the bleached finger coral with their polyps out at night. This is not typical during the day. They do feed at night, but just an interesting picture nonetheless. So now that we know the types of coral and the severity of the bleaching, you can start to get more specific with your reporting. So if you have your slate, you would tally the colonies, but if not, you can just summarize and kind of keep it in your head. When you go to report online, you'll be able to submit an observation of exactly as you've tallied them here, but using more of a categorical scale, so we don't need the specific numbers. It's just going to be single, few, and many, and we'll go through that in the next slide. The categorical scale, again, just makes it easier, just in case you forgot the slate, you didn't count exact numbers. We all know how diving can be. Moving forward, this is exactly what your form looks like online. So you can indicate, again, single, few, or many, and that's for each type of coral. So we're going to go through a couple rounds of practice for brain and mound coral. Brain, mound, boulder, coral, since those are the two most type that you'll probably see out there. We're going to start with the brain corals first, from left to right. So we have 3 brain corals that we saw with no stress, let's say. We all went diving together. We saw 3 brain corals, no stress. So we're going to mark those in the few category. Right here. Now for paling, we saw 6 colonies, let's say. So that's going to go into the many, as you can see here. Now let's say we didn't see any partially bleached brain coral. That one's going to get marked as none. And then finally, we saw 1 brain coral that was completely bleached. So we're going to mark that as single. We're going to move forward to the mound, boulder corals, and we're just going to run through another practice round. And again, this is going to get easier if you use the data sheet or the online reporting or your dive slate. Again, from left to right, on our slate, for example, we all went diving again. We got lucky, 2 tank dive. We saw 5 mounding coral with no stress. So that one's going to get marked as many. Let's say we saw 1 paling colony, so we marked that one as single. And let's say we saw 2 colonies that were partially bleached. So that one's going to get recorded in the few. And let's say we saw no fully bleached corals. We're going to mark that as none. So before I move forward, do we have any questions about the bleaching information before we move on to the disease section?
There were a couple, I just want to make sure, did we address any of the other questions that came up about like the hurricane activity and such?
No, I didn't see those questions. Sorry, I'm struggling to see the question.
Yeah, it's a small pain, but yeah. So did we answer the question about how quickly disease can move among colonies?
No, we didn't. So
okay,
that
yeah.
Good.
No, it's all you.
So the quickness of the disease spread, it depends. Depends on the disease, and it depends where it is. There's a variety of factors. It depends on the current, what type of disease it is, how susceptible the corals are around it. So there's many different factors. I wish I had a better answer.
Yeah, and so with the Stony Coral tissue loss disease outbreak, it spread a couple 100 meters per day at the beginning of the outbreak and moved particularly fast when it got to the Keys.
There you go. All right.
Then the next question was just with increased hurricane activity in the Gulf, does that cause higher levels of bleaching?
So not necessarily. Hurricane activity isn't directly correlated to coral bleaching. Now, the warmer water, which could cause more hurricane activity, may cause higher levels of bleaching, but not a definite. Those, you know, sea surface temperatures are a pretty good metric, degree heating weeks and stuff like that. So to answer your question, it may be just the higher water surface temperatures. That's all I see. Is that correct, Rachel?
Yeah, that's correct. That was the last one.
All right. A couple more seconds for questions. All right, I don't see any more. And again, just feel free to ask your questions. We'll stop at the end as well, and there's going to be a couple more breaks for questions. So now I'm going to move forward into coral disease and how you all will report that. So it's a similar process for coral disease, marking each type of coral. And there's 4 different categories we have right now in BleachWatch for disease, which is black band, tissue loss, white, which is associated with the current outbreak, growth anomaly, and other. I'm gonna go ahead and dive into all of these. So black band disease is another fairly common disease here in Southeast Florida. It was actually the first coral disease to be reported in the Caribbean and the Florida Keys. As the name suggests, it is characterized by a dark band separating the living tissue from the recently exposed skeleton, as you can see right there. Now, the next one we have is tissue loss. It's most associated with the current disease outbreak, or Stony Coral tissue loss disease. This also includes other white tissue loss diseases such as white plague, white syndrome, white band. And it's just difficult to discern these types of diseases in the field. So we're broadly considering all of these to be white tissue loss disease. Next up, we have growth anomaly. It's another condition to be aware of, although not anything serious at this time. Just again, better for you to be aware of this condition, or if you see it out there, just in case there is an outbreak. We don't want you to be under the water and you don't know what it is. You don't want to feel like this is healthy. It'll look like this, kind of a protrusion from the rest of the colony. It can sometimes be a slightly different color. Just looks kind of odd. Next, we have other, which are things like dark spot disease, other questionable disease that don't necessarily fit into other categories. And what that's going to look like, well, all these are going to look like on your data sheet is just like this. And best way to do it is to use descriptor words to describe the disease. It's extremely helpful for us if you have your slate and you're able to take a couple notes underwater, such as the lesion pattern, the shape, or the color, especially if you're not super sure on what you're seeing out there. Don't stress too much about remembering the lesion pattern terms. This is just something to be aware of. We have single and multiple lesion patterns. You can take notes, it helps, and it just helps us to categorize what's going on out there. And lesion shape. If you notice anything particular of the lesion shape, it can help us if it's linear, irregular, circular, but again, not super hugely important. We're going to move through section D. We're looking at the average bleaching and disease across the whole site. This question actually is very important because it helps us understand the overall severity of bleaching in consideration of all the healthy coral colonies that you saw. What did you see most of? And we also have an added category of dead coral with algae. This is also extremely important because site-wide, we want to know if that's mostly what you saw, and we want to take note of that. Moving forward, once the tissue is gone or the coral is dead, algae will start to grow on its skeleton. That's sort of what this category is about. We don't know why the coral died, possibly because of bleaching or possibly because of other reasons. Either way, it is dead. Unfortunately, this category is really intended to capture incidents where bleaching or disease was so bad that the corals have begun to die. And the way the reefs have been looking these days, this is unfortunately something that you may be seeing. Just keep a lookout on that. Next question, we have the overall percent bleached. Again, this is across the entire site, so you're just going to check only one, and it's how much of the coral was bleached. This is kind of a better idea of what this looks like. So photo 1, we got no bleaching over on your left. Photo 2, right here, there's still a lot of color in the corals, but there's, let's say, about 31 to 50% that are bleaching in this picture. And 3, you may be thinking, wait, why is that 100? You still see a lot of brown, but that's actually not live coral tissue. It's actually the underlying rock, so you wouldn't mark that anything other than 100%. Next question, we're going into disease. Overall percent disease. Again, this is across the whole site, so you're only going to check one of these. And your best estimate's good, but we're going to try to understand it how we showed you in that last slide. And the last question in this section D is other bleaching observations, because it's not just corals that bleach, stony corals that bleach. There are important indicators of bleaching events that are not included in the stony coral category. These include, but they're not limited to, fire coral, palithoa, and soft corals, which is why we include these on the data sheet just for reference. Just need a circle if you observe bleaching on any of these anywhere while throughout your dive. We don't care if it's technically paling or partially bleached. Just circle it, something to be aware of. Usually one of the first things, and this is why we bring attention to this, to start bleaching is these non-stony corals. These are not stony corals, like I just said. They're rather hydrocoral, which are colonies of hydroids that secrete hard calcareous skeletons. And they also have staining polyps, so again, we don't want to touch them. They're encrusting as well, so don't confuse them with other types of corals that may have grown over. And they're usually a tan to mustard color with white tips at the edges of the skeletal structure. Next up, we have the palithoa species. There's zoanthids, another species that typically bleaches first before the stony corals. And if you look at the picture on the right here, on the left you have the palithoa. On the right, you actually have a great star coral. The palithoa, it's a little hard to see in this picture, actually has 2 rings of tentacles around their mouths, and they're often irregularly shaped. Now, again, palithoa, it's not a stony coral, and it's encrusting as well. Kind of similar to anemones. Once you see it first, you won't get confused again. And you can always add pictures and say you don't know. That's totally okay. We look through all the reports and verify them. So again, you don't have to be experts at this just yet. This is another picture. This is actually at Lauderdale by the Sea during the 2014 bleaching event. Moving forward, we have Gargonians. Usually there's the last organism to bleach, so this is a good indicator of a severe bleaching year. There's many different species: sea rods, sea whips, plumes, sea fans, and there's 2 encrusting species. Moving forward, we have a couple examples of Gargonians with polyps retracted and extended. If you look on the left, the corky sea fingers have a purple rind, if you think about it that way, that extend. This is actually the extended polyps. They have a purple fuzzy appearance. It's on the left. And on the right, you have an encrusting Gorgonian that has more of like that tan rind. If you think about it like that, extended polyps. They look like blonde flowing hair, I guess. That's the way to look at it. Next section, last section, is your notes section. And you're going to record any other observations you see, invasive species, marine debris, or anything else that you see out there that you think we should take note of. As much detail as you can provide helps. For example, specific species names or other marine incidents, debris, lionfish, whatever you see out there, we want to know. So where are you going to put it? You're going to send it into cfan.net/bleachwatch. The whole point of taking this training is to compile reports. So once you're done, don't forget the most important step, which is submitting your data to us. So the data is good. Sending it to us makes it good. Now, when you're done filling this out, just remember sending it in, even if there's no data or even if there's no bleaching. Just want to emphasize that if there's no bleaching or disease, we still want to know, because we are gathering as much data as possible to give a health assessment kind of the reef, so we know what's going on out there. Even more important, at least to me, are pictures. Pictures are the most helpful thing to add to your report if you have an underwater camera. The online report form actually allows up to 10 photos, depending on the file size, but you can always email them directly to us. And again, I'll put my email at the end, and make sure they are sharp, in focus, and white balanced. So you can see that photo in the left, very, very difficult to see. I can zoom in, I can edit it, and do my best, but please, please, please make sure they're sharp, in focus, and white balanced. It's not very helpful if they're not. Make sure to take a picture of the whole colony and then another photo up close of the polyps or the lesion. And then all the information we get from you eventually is combined with all the environmental monitoring data that we talked about earlier, which will help produce our current conditions reports. And by taking this class, like I said, you will receive all of these, and we'll also make them available online. With the goal being, we're providing information regarding the current conditions to effectively communicate and raise awareness about mass coral bleaching events or whatever is happening out there. And remember, just sending in your photos, they can be included in the current condition reports, and we'll make sure to credit you, don't worry. And who knows, maybe you'll get famous off your wonderful photography. Moving forward, part of the bleaching response plan for Florida's coral reef, these current conditions reports are sent out monthly during the bleaching season, which, if you don't know, is from July, which is coming up sooner rather than later, surprisingly, to November, when the risk of bleaching is the highest. But if environmental conditions are favorable for bleaching, such as elevated sea surface temperatures and low winds, then the reports are sent out every 2 weeks. A weekly report has not occurred yet, crossing our fingers, mainly because it's been difficult to get enough observer reports in that frequently, so there's really nothing new to report. You can also check us out online. It's going to look just like that. It's at www.cfan.net/bleachwatch. It's going to have all the resources, everything we talked about today, historical current conditions reports, all the, like you can see, the bleaching fact sheet, disease fact sheet, data sheet, your data sheet instructions, coral condition ID guide, and I believe we also still have our PowerPoint on there. So you can review the information at any time. Now, our online report form, this is what it looks like. You may have taken this course before. I haven't asked, so If you've taken it before and you've gone on the BleachWatch before, it may look a little bit different, and that's because we switched some of the look of this, but it's all the same information if you've ever gone before. And before we would jump into the review questions, which is going to be the last part of this training, I wanted to also mention that there are additional opportunities to further your knowledge. So you're going to take this, or by taking this virtual classroom training, let's call it, you're now eligible to participate in any in-water training. In-water trainings, they typically do a classroom portion in the morning, which is the same you're taking right now. It's completely free. And there's times that we'll do an in-water portion, which is a 2-tank dive. Typically, there's some costs associated with them, just with the charter. But just want to bring that to your attention. And whenever these are scheduled, we'll be sure to pass that information along. And if you would like to take this even further, you can become a BleachWatch instructor, just like me, to be able to teach this class to others. Instructor workshops include a full day of classroom training and a half day of in-water training. Right now, we're not sure when these next classes are but as soon as they're scheduled, we're going to make sure that we share them through all of our channels so that you all know when it's happening. Finally, just want to conclude today's presentation before I get into the review, a short video. Some of the other ways you can help to protect Florida's coral reef, in addition to getting out there and submitting BleachWatch reports.
Florida's coral reef is purely Florida. Abundant with natural beauty and marine life, Florida's coral reef welcomes millions of visitors annually and provides vital economic support for coastal communities. You can help protect this natural wonder by always properly disposing trash and recyclables, choosing reef-friendly products like mineral-based sunscreens and sustainably sourced seafood, never anchoring, touching, or standing on reefs, and supporting local coral conservation efforts. Learn more at floridascoralreef.org.
Awesome. So thank you so much for tuning into this class. I'm going to be sticking around just to answer some questions, going through some review photos of different types of corals, because I knew we kind of blew through that, different conditions. So if you want to stick around, get a little more practice, you're welcome to do so. If you can't stay, I know we're all busy. That's totally okay. I tried to be efficient with the time. I respect all your time. And if you're curious about any other ways to get involved in the future, check out cfan.net, Southeast Florida Action Network, member our citizen science monitoring and reporting system. And these extend, again, through Martin County all the way down to Miami-Dade. Thank you. Again, my name is Tyler Mominey, and you can reach out to me at my personal email down there below. Thank you, everyone. All right, I'm sticking around if you guys got the time just to do some review, and if you have any more questions as well. All right, if it lets me change slide, there we go. So I'm just going to show a series of photos and give you some time on each photo. I don't have these in the poll, so don't even worry. This is just kind of for review between us two. And yep, just note the type of coral and condition, and then we'll review each photo, and I can answer any questions you might have about the answers. All right, the first photo here. So again, we're going to try to categorize it at least by what category it is. Flowering cup, maybe it's rain coral, maybe it's Mound Boulder. Give you a couple of seconds on that. Hey, look at that. We got Boulder, yes. Can't really see who that was, but yes, it's Boulder. You can tell because it's kind of encrusting over what it's growing, over. And this specific coral is a smooth star coral, and it's paling. You can see that from the splotchiness of the bleach tissue. There's still some healthy tissue there. I'll move forward to the next one. So note again what type coral this is, if you know the species, and what the severity of bleaching is. All right, this one is a brain coral. You can note from the brain-like grooves, that kind of gives it away. This specific brain coral is a Boulder brain coral, and it is partially bleached, as you can see from the patches of white tissue here and lots of healthy tissue here. And there's actually some Palithoa on the left side. Next one we got here. Remember to note what type of coral, species if you know it, and if this is bleaching or disease. All right, so this is a Mound Boulder coral. This specific one is a great star coral. And yes, correct, it is disease. You can tell specifically from this dead with algae, there's that specific kind of lesion pattern. And the specific disease is tissue loss, white. Next up, this one is kind of a trick question. It's not necessarily a stony coral. So if you know the answer to this, so just note if it's a disease or if it's bleached. And for this one, you would just circle on your data sheet. So this one is Palithoa. So not a stony coral, but you may see this out here. And yes, correct. This is a bleached non-stony coral. So for this one, you would just circle it on your data sheet. Make sure to report that as well. Moving forward, we have two corals actually, so kind of a trick question. So note what type coral it is, and if it's bleached, not bleached. So for this one, the two corals on the left, it's a Mound Boulder coral, specifically a massive Starlight Coral. No stress, so this is what we want to see. And on your right is the Flowering Cup Coral, and it's specifically a smooth flower coral. And again, no stress, so this is what you want to see out there while you're diving, swimming, snorkeling, whatever you're doing out there. Next up, type of coral, and for this one specifically, if it's diseased or bleached, and if you know the disease or level of bleaching. All right, so this is a brain coral. It's actually a knobby brain coral. You can see again with those grooves in there. Right here, kind of looks like the brain. And this specific is black band disease. You can see the kind of dead skeleton in the middle with some algae there. But it's also paling. I know it's kind of confusing, but what we would want you to put here is that there's black band disease because it's actually a terminal condition. All right, we got a couple more here. I'm gonna blow through these. So there's two in here. Type of coral, whether it's bleached or not, and the severity of bleaching. All right, on your right, we have a leaf plate sheet coral. Specifically, it's a lettuce coral, and it is fully bleached. And on the left, I know this one looks a little odd. This is a Mound Boulder coral. It's a massive starlight coral, and it is partially bleached. Even though it's that purplish color, again, that's just kind of the pigment color, but it is partially bleached. Next up, type of coral and how you would mark it on your sheet. All right, this specific one is a Mound Boulder coral and a great star coral specifically. And this one would be marked dead with algae. There's no tissue, and you can see here it's starting to turn green. So that's an important observation to make. Make sure that you're reporting that to us. Next up, we'll move through these pretty quickly. Only got a couple more. All right, this one is a brain coral, specifically a grooved brain coral. And this one actually is diseased. It's tissue loss disease. And if you look here, you can kind of see portions of the bare skeleton and then the portions with the tissue. Very hard to see, kind of depends on your monitor. But yes, an important distinction between bleaching and disease is the lack of tissue for disease. Versus bleaching, still going to have that tissue even if it's translucent. All right, next up for you, we got 5 more here. All right, this specific one is a Mound Boulder coral. You can see by that kind of encrusting and mounding shape. And it's a mountainous star coral, and this one's actually healthy. It may have early phases of paling, but it's also pretty common for the tips of the knobs to be lighter in color for the mountain and boulder corals. Next up, this one's kind of a trick question. Type of coral and what's happening to it. All right, this one specifically is a Mound Boulder coral. I just want to point out here in this circle, that's predation. So you can see kind of those bits taken out of it, not necessarily linear, just look a little odd. So that one is specifically predation. Next up, only got a couple more. What do we got here? We'll give it a couple seconds. All right, here we have a Mound Boulder coral. This specific one is a Mountainous star coral, and this one is partially bleached. So note kind of these upper portions of the knob, they're bleached. And it's interesting to note they're just the ones that are exposed to the direct sunlight. Next up, give this one a couple seconds. All right, this one is a branching coral. This is actually a pillar coral, and it's actually diseased. This is tissue loss. It would be marked as tissue loss white. And kind of notice here that green area that's been taken over by algae.